A scientist walked into his laboratory one morning, sat down at his desk, and started tickling a rat. Not because he was bored, not because he was strange, because he had a hypothesis that nobody in the scientific community wanted to take seriously. He believed the rat was laughing, and he was right. His name was Panksepp. He was a neuroscientist at Bowling Green State University, and in the late 1990s, he discovered something that should have changed the way every human on the planet thinks about themselves. When he tickled the rats on the back of their necks, they emitted a sound at exactly 50 kilohertz, too high for the human ear to detect without equipment. But when his team slowed the recordings down and dropped the frequency, the sound that came out of the speakers was eerily, unmistakably familiar. It sounded like giggling. Before we get to why rats laugh, we need to talk about a number. 97. That's the percentage of human genes that have a direct counterpart in the rat genome. Not similar, not related, a direct counterpart. The same gene doing the same job in a completely different animal. In 2004, an international team of scientists, led by the Baylor College of Medicine in Houston, completed the full genome sequence of the rat. The project cost over $118 million. It was funded by the National Heart, Lung and Blood Institute and the National Human Genome Research Institute. When they published their findings in the journal Nature, the conclusion was uncomfortable for a lot of people. Rats and humans share approximately 90% of the same genes. Almost every single disease gene in the human body has a rat counterpart. Every organ system, cardiovascular, nervous, digestive, reproductive, operates by nearly identical mechanisms in both species. The researchers found 280 large regions of sequence similarity distributed across the chromosomes of both animals. They called them syntonic blocks. What that means in plain language is this: the same ancient instructions, written in DNA over millions of years of evolution, are running inside you and inside the rat at this exact moment. You didn't come from rats, but you both came from the same place. And whatever that ancestor was, it built something that worked so well that evolution didn't bother to change most of it. When his paper came out in the year 2000, published in the journal Behavioural Brain Research, the title alone made other scientists uncomfortable. It was called 50 kilohertz chirping in response to conditioned and unconditioned tickle-induced reward in rats, effects of social housing and genetic variables. The parenthetical in the title said, laughter? The question mark was not accidental. Panksepp knew what he was claiming, and he knew the resistance it would face. Here is what he found. The rats didn't just emit the chirps when tickled, they sought out the tickling. After a few sessions, the rats would actively run toward the researcher's hand, the specific hand that had tickled them, and position themselves to be tickled again. They were asking for it. The rats that chirped the most were also the most playful. They were more likely to initiate social interaction, more likely to bond with other rats, and, this is the detail that Panksepp found most significant, more likely to approach unfamiliar situations with optimism rather than fear. In follow-up experiments, his team found that tickled rats pressed a food reward lever far more frequently than a shock avoidance lever when presented with an ambiguous sound. They interpreted uncertainty as possibility rather than threat. In 2018, researchers at Humboldt University in Berlin confirmed and extended Panksepp's findings. They found that rats not only laugh when tickled, they anticipate the interaction. Their brains showed measurable activity in regions associated with positive emotion before the tickling even began. The laughter wasn't a reflex, it was an expectation of joy. In 2011, a neuroscientist at the University of Chicago named Peggy Mason ran an experiment that nobody expected to go the way it did. She placed a rat inside a small restraining tube. The tube could only be opened from the outside. Then she placed a second rat, a free rat, in the same arena. The free rat had never seen the tube before. It had never been trained to open it. It had no incentive, no reward waiting. Within a few sessions, the free rat had taught itself to open the latch, every single time. But that wasn't the part that surprised everyone. Mason then placed 2 containers in the arena simultaneously. One contained a trapped rat, the other contained 5 chocolate chips. The free rat had a choice: help a companion or eat chocolate. The rats chose to free the trapped rat first. Then, and this detail is the one that keeps appearing in scientific papers years later, they shared the chocolate. The study was published in Science magazine, one of the most prestigious journals in the world, under the title Empathy and Pro-Social Behavior in Rats. Jean Decety, the co-author and one of the world's leading researchers on the neuroscience of empathy, said this: "This is the first evidence of helping behavior triggered by empathy in rats. All of this tells us that acting on empathic feelings to help another in need is a biological, and in fact a neurobiological, mandate. It's in our brain. Not our culture, not our upbringing, not our religion, our brain. The same brain structure that drives a human being to stop and help a stranger on the street drives a rat to open a latch for a trapped companion. It evolved before language, before cities, before the concept of morality existed. It was just there. Built in. Here is where it gets uncomfortable. In follow-up experiments, Mason's team discovered something that made the empathy results more complex. The rats only freed trapped companions if they had previously socialized with a rat from that same group. Strangers got left behind. When the free rat was placed with a trapped rat from an unfamiliar strain, one it had never interacted with, the helping behavior dropped significantly. Familiarity bred empathy. Unfamiliarity bred indifference. And when scientists administered midazolam, an anti-anxiety medication, to the free rats before the experiment, something remarkable happened. The medicated rats stopped helping, not because they couldn't open the latch. When the tube contained chocolate instead of a trapped rat, the medicated rats opened it without hesitation. The drug hadn't affected their ability. It had affected their feelings. Without the anxiety of watching a companion in distress, there was no emotional drive to act. Peggy Mason's conclusion was direct. Helping others is good for the species. It's a mammalian trait that developed through evolution. Here is what nobody in the coverage of these studies bothered to point out. We spend enormous energy trying to define what makes humans unique. Language, consciousness, morality, empathy, love. And one by one, science keeps finding those same things in animals we had dismissed. The rats laugh. We said, that's not real laughter. The rats help. We said, that's instinct, not empathy. The rats share chocolate. We said, that's conditioning, not generosity. But the neuroscience doesn't make that distinction. The same brain circuits that fire when a human feels joy, the dopamine pathways, the limbic system activation, fire in rats under identical conditions. The same circuits that activate when a human witnesses someone suffering and chooses to help. The anterior cingulate cortex, the insula, activate in rats watching a caged companion. The brain doesn't know it's supposed to be a rat. It's running the same ancient program. And here is the part that the scientific community is still quietly debating. If the rats only help their own group, if familiarity is required for empathy to activate, then rats aren't just like us at the neurological level. They're like us at the social level too. In-group compassion, out-group indifference, the rat that shared chocolate with its friend and walked past the stranger. Sound familiar? Panksepp spent the last decades of his career arguing for something he called affective neuroscience, the study of emotional experience across all mammals. He believed that joy, fear, grief, and play were not human inventions. They were biological infrastructure, built into the mammalian brain long before humans existed. He died in 2017. His tickling experiments are still cited. His hypothesis about rat laughter is now considered mainstream neuroscience rather than fringe speculation. But the deeper implication of his work is one that most people are not ready to sit with. If the capacity for joy is ancient, if empathy evolved before language, If the instinct to help someone you love was encoded into mammalian DNA before the first human ever walked the earth, then what you feel when you laugh with someone, when you stay up all night with a friend in pain, when you feel an irrational need to help a stranger, that's not civilization. That's not religion. That's not something you were taught. That's 80 million years of evolution doing exactly what it was built to do. The rat in Panksepp's laboratory wasn't imitating humans. We were never ahead of it. We were just the same thing, in a different body, telling ourselves a different story.